Sir, please start.
Good evening, everybody. A warm welcome to all of you, our dear doctors, international speakers, guest panelists, and all my viewers. A warm welcome on behalf of Biotech in this global webinar, Eyes and COVID-19. And just imagine who amongst us ophthalmologists is a very special guest today evening, and he's none other than Professor Dr. Murat Akowa. Let me take the honor to introduce this gentleman. Dr. Murat Akowa is a professor of medicine in the infectious diseases in the department at the Hacettepe University School of Medicine in Ankara, Turkey. He was the president of ESCMID and currently serves as a vice president of the International Immunocompromised Host Society. Dr. Akowa's research interests include infections in immunocompromised host and antimicrobial resistance. It's a great honor to have you, sir, amongst us and to share this platform, shedding vital information regarding this notorious COVID-19 disease caused by SARS-CoV virus. All over to Dr. Murat Akowa.
Thank you very much, Dr. Wahi, for this nice introduction. I'm very pleased to be here and to attend this session. And I also, my special thanks to Biotech company, who is facilitating this webinar. And thank you very much again for making it possible for me to be here. Today, what I'm going to do is actually, during the following half an hour or so, try to give you an update about the disease itself and some emerging clinical experience. So let me start with the basic terminology. I mean, I'm sure all of us is very familiar with this one, but just to remind once again. So there are two definitions, actually. The first one is the COVID-19, which stands for Coronavirus Disease 2019, indicating that the disease was described in 31st of December, actually, 2019. And the COVID term is derived from the coronavirus disease. Then the virus itself, which is named after SARS coronavirus, because the SARS coronavirus, which was originally described in 2002 and caused epidemics in the Southeast Asia and elsewhere, a serious respiratory syndrome. And this virus is also a single-stranded beta coronavirus like this one. And between SARS coronavirus, the original one, and this one, SARS coronavirus 2, shares 82% of genetic similarity, but their special enzyme, which is RNA-dependent RNA polymerase, has similarities 96% between two viruses. Well, since first description of the disease, early January, there has been an explosive number of publications in the medical literature. And this slide, just from last week, from the Economist Journal, and it shows us that there are now more than 7,000 papers either published in a peer-reviewed journal, which is approximately 4,000, and there are more than 3,000 preprint papers which have been uploaded but not peer-reviewed yet. So you can imagine how fast the reaction from the science to this disease, and there have been enormous numbers of publications. So the knowledge is changing. I mean, yesterday we had a rehearsal, for this meeting, and since yesterday there have been lots of new information. So I'm going to try to include some of these information into this talk as well. Well, let's talk about the coronaviruses in general first. I mean, these viruses are not only pathogenic for humans, but there is a wide variety of animals as well which can be affected by these viruses. And among them are camels. Camels is the intermediary hosts for the MERS, which is another coronavirus disease, beta coronavirus disease, cows, cats, and bats. And actually this coronavirus, SARS coronavirus 2, again has very close similarities between a special bat virus, which is endemic in the Southeast Asia. Well, the regular coronaviruses actually causes up to 30% of upper respiratory tract infections in humans. But as I mentioned earlier, now there are three human coronavirus diseases which are severe. The first one is SARS, the second one is MERS, which is still going on, and the last one, of course, is the SARS coronavirus 2. Now, this is the actual number. I prepared this slide just approximately 3.5 hours ago from a Johns Hopkins University web server, which is important in here. Now, there are more than 4,300,000 cases all over the world. US is leading, but what is emerging at the moment now, the number of deaths in United States, which is over 84,000, exceeds the number of actual cases in China. So there are more dead people in the United States than the actual cases in China. Russia is another place where the pandemics hit worse. So they are second on the row, and then United Kingdom, Spain, Italy, Brazil, etc. Turkey, my country, is on the 9th of this rank. Now, a bit of information about the virus itself. So this is a schematic representation of the SARS coronavirus, either 1 or 2. It's, as I said, a single RNA virus. There is an envelope covering this single-stranded RNA, and then there are a variety of proteins getting out, protruding from this envelope. The most important one is, as shown here, is the spike protein. Spike protein is the means of entry into the cell, actually. So what happens when the virus is exposed to human cells? First, it gets activated from a protein, a protease, actually, which is again present on the host cell membrane. This is called TMPRSS2. And then once the spike protein is activated, then it attaches to its receptor on the cell, which is angiotensin converting enzyme 2, which is again a protein widely distributed on human tissues. And this is the distribution. This is a paper which was published in Nature Medicine, 28 March this year. So it shows us a large variety of tissues in the human body expresses both angiotensin converting enzyme 2 and also this protease, the second column here. And the third column indicates the co-expression of these two factors. So if you go to, for example, nasal cavity, ileum, colon, lung parenchyma, and also cornea is a place where these two factors are co-expressed. Let's have a look closer to the corneal tissue. So as you see here, the co-expression is present in superficial conjunctiva, limbal superficial cells, which indicates that the virus can penetrate through the cornea or conjunctiva to infect the human beings. Now, this is again a very basic slide. It's like from a biology textbook from a high school kid, but it shows us the comparative sizes of the cell. Well, now the coronavirus, the SARS coronavirus 2, the size is approximately 120 nanometers. This is 1/10 of a classical bacterium, which is E. coli, and the E. coli is 1.2 micrometers, so it's 10 times bigger than the virus itself. Why I'm showing this slide? It's because of the viruses are usually, and this virus particularly, spread through droplets or aerosols. When we say droplet, it is bigger than 5 micrometers, and when we are talking about aerosols, it varies between 300 and 5 micrometers. So the aerosol is less than 5 micrometers. And the aerosol, the important thing is, once the aerosols are creating, either we are shouting, singing, sneezing, or coughing, we produce a lot of aerosols. And these aerosols can hang in the air for a while, and I'm going to come to that. Now, here is the mode of transmission. As I already said, it's either droplets or aerosols. And once these droplets and aerosols are produced, they contact to oral, nasal mucosa, and conjunctiva, or contaminate the surfaces, and then it can disseminate the disease. The important point here is that the asymptomatic people can also produce viruses, shed viruses, and from their secretions, the virus can be transmitted to the other people as well. Now, here is a study which was published in JAMA, 26th of March this year. It examines the turbulent gas clouds produced by a person who was sneezing. So on the picture, you see once someone sneezes, it creates a cloud of aerosols and droplets. And among these clouds, there are a variety of droplets and aerosols, varying sizes. And these droplets and aerosols actually can reach up to a 8-meter diameter distance. Well, this is important because, you know, we have a social distance concept saying that between 1 to 2 meters, you have to protect yourself. So you don't have to get closer less than 2 meters to someone who might be infected. That might not be true or enough, because if you are within the diameter of this 8 meter, actually you can get affected from a person who is sneezing or coughing or shouting, for example, because those droplets actually can reach to that distance. This is important. And then the attack rate could be very high. This is a paper just 2 days ago, published by the American CDC. It's the Morbidity Mortality Weekly Report. And it shows us that one symptomatic person can infect a large group of people. And in that case, it was a choir practice, and one person was attending this choir session for 2.5 hours. And then he infected 87% of the group, the whole group. And among them, 2 of them unfortunately died. So the efficacy of transmitting disease might be very, very high with those infected patients. Now, the second issue is the aerosols and the surface stability of the virus. This paper was published in New England Journal of Medicine in March, and it shows us the aerosols, so those small particles, which is smaller than 5 micrometers, can hang over in the air up to 3 hours on this one, the upper row on the very left-hand side. The relatively safe materials are either copper or cardboard. In the copper, the relative half-life is around an hour. So up to 4 hours, you can detect viable virus. The cardboard is up to 24 hours, but in steel and plastic, it might be either 48 or 72 hours. So on these materials, if it's not cleared, if it's not disinfected, the virus can stay up to 48 or 72 hours. And of course, someone touching on this surface and then putting his or her hand to mouth or nose, he or she can infect himself or herself. Another important point is the aerodynamics of these aerosols. So this study is coming from China. What they did actually, they examined in 2 hospitals, various places, collected aerosols, and looked at the virus RNA in these aerosols. So what they detected in the isolation wards, in well-ventilated patient rooms, and not crowded public areas, the viral RNA concentration is very low. But if you go to the patient restrooms and the common areas for medical staff and crowded public spaces, there is very high rate of viral RNA in these aerosols. And as I said, these aerosols are hanging in the air at least up to 3 hours. And in these places, the cross-contamination could be very high. This information should have very important implications. Then the issue is, of course, the face masks. I mean, face masks, unfortunately, have not been readily available, these surgical masks in many countries, including United States and many Western countries. So it was a big hurdle for people to obtain these masks and protect themselves. But actually, the question is whether these masks are protective. This paper in Nature Medicine examined the coronaviruses, not specifically coronavirus 2, SARS coronavirus 2, but the regular coronaviruses, and influenza viruses and rhinoviruses. These are the common causes of upper respiratory tract infections. And they found that if you are using a face mask, these surgical face masks could prevent transmission of these viruses. Then there is this paper. This is a chemical society, American Chemical Society Nano Journal. I mean, it's not a regular medical paper, but they did a very interesting analysis. So they tried in the laboratory conditions various materials for preparing cloth masks, and they have very interesting results. So first of all, they determined the best materials for protection is cotton with tight waves, silk, particularly if it's used 4 layers. So if you have a silk scarf, for example, if you fold it in 4 and then use it as a face mask, it's highly protective. Then chiffon and flannel. The ladies should know about these material because this is, you know, very, very popular, I guess, in the ladies' clothes. Then you can reduce the efficacy of transmission up to 90%. However, one thing is important. If you put these masks improperly on your face, that can reduce the filtration efficacy by 60%. Here it is. And then you can use these materials in combination. For example, the green one, cotton silk, cotton chiffon, or cotton flannel. And as you see here on the graph, these combinations are as effective as an N95 mask. So you don't need to get a high N95, but you can use this one. But of course, N95 is only for medical personnel, not for the regular people. And the lower row indicates the gaps, the unfit of these masks. And as you see, the lower part of this graph, if these masks do not fit properly in your face, so there are gaps between the mask and the face, the efficacy drops below 60 or 40%. So that's another important piece of information. On the other hand, this is the actual hospital setting. This paper in Annals of Internal Medicine, 6th of April, from 2 hospitals, 4 different patients. They checked with patients coughing with a mask, with a surgical mask, with a cotton mask, and without mask. And unfortunately, they couldn't find any difference between patients wearing a mask or without wearing a mask. So the efficacy was not very good. The previous paper was in the experimental situations, not the actual patients or people wearing masks, but these are the masks. So there is still confusion between this mask issue. Now, another question is the duration of contagiousness. So how can a person infected with SARS coronavirus 2 can transmit the disease? Well, actually, the transmission starts between 2 to 3 days before symptoms, which is important because that means a lot of asymptomatic people around us and can shed the virus, can infect the other people. And the estimate is that up to 44% of patients actually get infected from those asymptomatic people. This is again very important. And this then underlines, emphasizes the importance of wearing a mask when you are present in a public place, particularly in a crowded place, in a shopping mall, for example. However, within a week of symptom appearance, the viral shedding and the rate of transmission will decrease gradually. So after a week, probably the transmissibility is limited significantly. But in isolated cases, it's been described that up to 37 days since the symptoms start, the viral shedding can still be detected. But of course, this is viral RNA, so nobody knows whether it's a live virus or not. Well, we are aware of the classical respiratory symptoms. I mean, it's cough, short of breath and fever. But now what is emerging is that there are a variety of non-respiratory symptoms and signs. And of course, today's topic is eye. So there are lots of eye symptoms which are going to be addressed by the other speakers. But here you see that diarrhea, anosmia or dysgeusia, several neurological findings, including stroke, various cardiovascular signs, myopericarditis, arrhythmia, hypercoagulability, and emboli can frequently be found in those patients infected with SARS coronavirus 2. So this is important and this is emerging. And I didn't prepare a slide, but we have been discussing before this session, and now it's another emerging issue that the disease is not sparing kids. There is a syndrome now described particularly in United States and United Kingdom, not only small children, but also in adolescents as well, up to 16 years old. There is a syndrome very similar to Kawasaki disease, which attacks heart and coronary arteries, and also cause high fever, lymphadenitis, and skin eruption. And there has been a mortality approximately 68% among those kids. So this is something new and very scary as well. Well, what we know at the moment, the disease causes significant endothelitis. So it attacks endothelium. Well, this is obvious because endothelium is very rich angiotensin converting 2 receptors. And here I show you three different slides. The one on the left-hand side is from the glomeruli and the tubular endothelium from kidney. So the arrows indicate the viral particles and also the inflammation by mononuclear cells. The medium portion is from the intestine. Again, there are viral particles and inflammatory lesions. And on the right-hand side, we see alveolar and pulmonary arteries, again, infiltrated by the virus and the inflammatory cells. So in all these tissues, there have been viral particles. That means these tissues have been attacked. So it can explain the pulmonary and non-pulmonary symptoms. Now, about the detection of the virus, the standard test is the polymerase chain reaction, reverse transcriptase polymerase chain reaction. And in the typical clinical setting, you get a nasopharyngeal swab and then do a RT-PCR. But unfortunately, if you see here in nasal swabs, the sensitivity is approximately 60%. It can up to 90% if you have a bronchoalveolar lavage fluid, but this is very rare, of course. This is an aerosol producing procedure, and it's very rarely it would be attempted by the intensivist, but we use nasal swabs. And the nasal swabs, it seems not highly reliable for exactly detecting the virus. Now I'm coming to the end, and I will say a few words about the therapeutic agents and modalities. Unfortunately, so far, there has been not a single drug which has been shown 100% effective for treating the disease. Remdesivir, which is going to be addressed by another speaker today, and other viral RNA synthesis inhibitors, lopinavir, ritonavir, and anti-HIV drug. There is a favipiravir from China and in my country, Turkey, which is widely used in these 2 countries. Then antiviral entry inhibitors, chloroquine, hydroxychloroquine, IL-6 inhibitors like tocilizumab or interferons. And now there have been clinical trials about convalescent plasma transfusion and also mesenchymal stem cell transplantation. Well, of course, the vaccine issue is a hot topic. There have been more than 100 candidate molecules for a vaccine. But in this slide, you see the most promising ones. There is one from University of Oxford using an adenovirus vector. And there is another one from NIH and a company in the US called Moderna. They are using mRNA nanoparticles. And this one, the last one, now is now moved forward to a second phase. So the first phase was successful. But unfortunately, we are not expecting an effective, I'm saying quotes, in quotes, an effective vaccine within a very short period of time. So probably in September, October, we are going to have more information, but widely available vaccine would not be available within a year or so. So it's a distant hope at the moment. Now, before ending my talk, I'd like to say just a few words about the future of this pandemic. This slide explains the 3 scenarios proposed by a report which was recently published by University of Minnesota and a group of people called CIDRAP. And it shows us the virus can go peaks and valleys. like that shown in the scenario 1, or can create a fall peak in September, October, or now the peak ends right here, but then ups and downs as you see here. And all these scenarios, depending on the preventive measures taken in the society. So if those lockdown measures and social distancing can be maintained, the lower levels of these peaks will be attained. But otherwise, we are going to experience either of these scenarios until 2020. And finally, everybody agrees that the fate of the disease will basically depend on the herd immunity and availability or non-availability of an effective vaccine. But in both scenarios, the disease probably will go into a stage like an influenza-like seasonal illness. Well, of course, the duration of the immunity, the natural immunity or vaccine-produced immunity will be critical here. If the immunity will last less than a year, and this is typical for all other coronaviruses, we are going to experience yearly epidemics. If the immunity is approximately 2 years, then the epidemic will be every other year. But if, fortunately, we don't know, the immunity will be so-called permanent, like in measles or polio, then the disease at least will disappear for 5 or more years. But we are yet to see, and we don't know how long will last this immunity. So I think I'll stop here. Thank you very much again for giving me this opportunity, and I will entertain any questions you might have at the end of session, I think. Thank you very much.
Thank you, Dr. Muratjakova. It was indeed a wonderful presentation, and I would just sum up that I was virtually feeling that I was just watching a movie on coronavirus. It was so well said right from the beginning till the end, talking about the finer details about the basics which are required, the precautions which are required, as well as the forthcoming problems which can be there, as well as all about herd immunity. So I really appreciate it. It was a very good and a comprehensive talk. And now I feel that Maria is there, so my job is over. And she'll be introducing Professor Yonka Akova, and then we'll be handling the session further. So all over to Maria. Maria, you have to unmute your mic. Maria, you have to unmute your mic.
Can you hear me now?
Yeah, all perfect.
Yes. Okay. Well, first of all, accept my apologies for Dr. Amsterdam. He had a really difficult surgery this morning. He's still in the OR. So I'm trying to call him, but I was not able to reach him. Hopefully he will be able to connect later during the webinar. And well, I should introduce myself. I don't know if someone did. I am Dr. Romero. I'm from Spain. I'm an ophthalmologist. I'm a specialist in oculoplastics and dry eye. And I'm going to introduce then the next speaker. It's Dr. Yonka Akova. I'm not sure if I'm pronouncing it well. It's kind of a challenge for a Spaniard to pronounce these names. So hopefully I did it well. And she is director of Department of Ophthalmology at Biotech Hospital in Turkey. And she's going to talk about COVID and the signs in the eyes and how ophthalmologists protect themselves during the visits. So, I give you the floor, so you can start whenever you want.
Thank you. Thank you very much for the kind introduction. I also thank to Biotech Global for giving me this opportunity to talk to you on ophthalmological findings in COVID-19 and how do we protect ourselves as an ophthalmologist and how do we serve in these days to our patients. COVID-19 and the ophthalmology link began at the beginning of this pandemic. This is a picture from Guardian newspaper showing 33-year-old Chinese ophthalmologist he is famous now, all ophthalmologists know him, who first warned about the COVID-19 pneumonia cases, and the officials accused him of making false comments at the beginning. Unfortunately, he died early February because of the COVID-19 infection. He was acknowledged later on, but he was the first one to recognize the disease. And there's another important finding about eye, which was published in February 6th at Lancet. A national expert of pneumology from Wuhan was infected during an inspection visit. He was wearing an N95 mask and he was thinking he was protected, but he did not have an eye protection, and he developed first conjunctivitis followed by respiratory disease. This is the first transmission through the ocular surface was published. How about the coronaviruses and the eye? This is a review from by Dr. C from Singapore. He especially evaluated the coronavirus ocular involvement in animal studies. Conjunctivitis, uveitis, retinitis, and optic neuritis has been shown to involve, has been shown to the presenting signs in animals. When we come to COVID-19, ocular involvement is mainly is in the form of conjunctivitis, but in 2 days before, there's a report in Lancet showing the retina involvement, which I will show later on. This is a paper including 1,099 patients from China with confirmed COVID-19 cases. Only 9 patients had ocular involvement in the form of conjunctival hyperemia. This is less than 1%. This is a retrospective study, and there's not a big description of ocular involvement. This is from February 28. There's another study for ocular findings of patients with coronavirus disease from Hubei, from China. This study included 38 patients with confirmed COVID-19, whom 4 had moderate disease, 2 severe, and 8 critical disease. 12 of these patients described as having ocular findings, it's almost 1/3 of the patients, in the form of conjunctivitis, conjunctival hyperemia, chemosis, epiphoria, and increased secretion. But only 2 patients had positive conjunctival PCR. This study was limited, because of the most of the patients were critical or severe disease, and the chemosis and conjunctival hyperemia might be related to patients being in the intubation related. There's another case from China, 30-year-old man with confirmed COVID-19 who developed bilateral acute follicular conjunctivitis 13 days following COVID symptoms. Conjunctival swab specimens was positive for SARS-CoV coronavirus at day 13 and 17, and this was similar to pharyngeal conjunctival fever. It was a mild follicular conjunctivitis. This patient was treated with systemic and local antiviral treatment. There's another case presentation from Canada. This patient had conjunctivitis and mild respiratory symptoms without fever, and his conjunctival swab specimens was positive for SARS-CoV coronavirus 2, and this case shows the high index of suspicion is important for the diagnosis. This is a big study from China describing ocular findings and proportion of conjunctival SARS-CoV-2 in COVID-19 patients, including 1,121 patients with confirmed COVID-19. Only 8 patients showed ocular symptoms. in the form of itching, redness, tearing, discharge, and forehead body sensation. This is 6.6%. But only one patient with severe disease had conjunctival PCR positivity. 2 patients without any ocular symptoms also had conjunctival PCR positivity. When we looked at the COVID-related conjunctivitis, mild follicular conjunctivitis with conjunctival hyperemia, chemosis, increased secretion is present in these patients, only 1 to 6%. Conjunctivitis may present in the middle or late phase of the disease, and transmission is probably with infected air droplets or aerosols, as mentioned previous talk, or direct contact from secretions finger to conjunctiva. This is the paper I mentioned. This is published. This paper was published in Lancet 2 days ago from Brazilian group. They described 12 patients. Actually, most of them there were hospital staff with COVID-19 PCR positivity and mild to moderate disease. When they did OCT and fundus examination, and this, as you see here, there's a hyperreflective lesions on OCT, here, here, and here, in the ganglion cell layers and inner layer of retina, as well as subtle cotton wool spots and microhemorrhages in the fundus. And this is the first paper describing retinal findings, and we probably will hear about this more. When we looked at the coronavirus in tears and conjunctival secretions, this is a study from China from February. 20 patients with confirmed coronavirus pneumonia, tear and conjunctival samples were obtained twice for PCR analysis, but only 1 patient with conjunctivitis had RT-PCR positive results. This is from Annals of Internal Medicine. It's a patient from Italy. Actually, this is the only case the virus was cultured. They were able to culture. This is the only case. And when they did the viral DNA detection, they found the viral DNA from day 3 to day 21. And day 22 to 26, there was no viral particles, but day 27, there was also again positivity of virus. And at the beginning, patient had conjunctival hyperemia and conjunctivitis findings, but after day 20, he did not have any sign of conjunctivitis. It's an interesting case. This is again related to infectivity of tears and viral shedding from a study from Singapore. They included 17 patients with COVID-19 with nasopharyngeal PCR positivity. Only 1 patient had conjunctival injection and hyperemia, but no sign of conjunctivitis. All tear samples were PCR negative at the 1 week, 2nd week, and 3rd week. And there was no viral PCR positive. All red ones were negative, showing negative results. This paper is already online, but it's very new information. Giving these results and the viral conjunctivitis and the tear containing virus, what we should do in the clinics. Of course, we know now all the physicians about triage, temperature screening, hand hygiene, and face masks at all times. But specifically in ophthalmological clinics, we need to wear face masks as well as protective eyewear or visors. We would like to see only our patients in the room. If possible, patients or caregivers should stay outside of the room, and slit lamp barriers, breath shields, as we call it, should be used in the slit lamps routinely. And as you see here, ophthalmologists and patients are advised not to speak during slit lamp examination because of the aerosol and the droplets. All equipment and environmental surfaces should be cleaned and disinfected between each patient. We do have many equipments. This is very important for us, as well as general cleaning. What we did during this period, routine eye care markedly decreased everywhere in the world, including surgery, lasers, spectacles, urgent ophthalmological visits, especially patients with sudden visual loss, blurred vision, visual field loss, red eye, pain, headache, eye injury, and patients with floaters are asked to come for examination. And we did a lot of telemedicine in order to understand which patients are urgent or not. And we did perform only urgent or emergent surgical operations, and intravitreal injections are generally performed. In terms of the hygiene in ophthalmology, hand hygiene and social distancing, of course, are very important, but also it's also very important not to touch and rub the eyes, and artificial tears and anti-allergic drops are helpful when necessary. We recommend wearing glasses more often than contact lenses if possible. If contact lens use is preferred or necessary, hand washing, good hygiene habits, and daily contact lenses are recommended. This is a paper from last week from David Park, American Academy of Ophthalmology CEO. What he describes, what is going to happen in ophthalmology, he says that we are not returning to normal. We are going to have a new normal. Especially what he describes in ophthalmology clinics, less waiting time, less crowded waiting area compare what we had before. Less people in the examination area, and personal protective equipments are very important. Masks, protective glasses, if possible, antibody testing of all staff, and telemedicine are new important tools for us. What we recommend in terms of seeing patients, we should ask our patients about fever, respiratory illness, sore throat, anosmia, or any COVID-19 positive family members before they appear in our ophthalmology clinics. And we should reduce number of seats in waiting areas, reduce number and duration of our visits. We need to make shorter visits with our patients. Patients and patient caregivers should wear face masks at all times. And in clinics, we need to have easy access to hand disinfectants and hand washing areas. How about surgery? How do we plan elective surgery? Preoperative PCR testing or antibody testing is becoming discussed in this area. It would be very nice if we could able to do the preoperative PCR testing all patients, but if we suspect anything, we should do the testing. For cases performed with sedation, surgical mask for patient during surgery is necessary. Surgeon should wear N95 mask if the patient is PCR positive and he needs to do, he or she should go surgery, or IgM positive. For cases requiring general anesthesia, N95 masks during intubation is necessary, and everybody else should be go outside of the room. And we use all the time 5% povidone iodine in our surgery. It should be also used in these patients, in all our patients as a surgical prep, since it inactivates all viruses, including COVID-19. And telemedicine is also maybe helpful for preoperative evaluation and postoperative follow-up when appropriate. Not all the time, but we can avoid some of the visits. In conclusion, ophthalmologists are one of the highest risk professionals in COVID-19, and personal protective measures are extremely important during examining and during surgery, and future safe practices will be shaped by protective measures. Of course, as mentioned previous talk, availability of safe vaccine and high level of public immunity. I thank you for your kind attention. I would be very happy to answer if any questions. Thank you.
Okay. Thank you, Dr. Yonka. It was a great review, actually. As an ophthalmologist, I should add, well, I don't know if you're doing the same in Turkey, but in Spain, our societies decided that maybe air tonometry was not considered the best to calculate, to measure intraocular pressure, because of the air could spread the ocular fluid, make like the droplets and aerosol in the air. I don't know if you guys are doing the same in Turkey, like avoiding.
Thank you for this comment. This is very important. There was a big discussion about the air pneumotonometers, air tonometers, if we should use them, if they produce aerosols during the examination. There's a big discussion, and we are not sure if they produce aerosols, but in this period, we avoid to use air tonometers. Thank you for adding this.
Okay, you basically. And well, as you said, since we are so close to the patient, we have to consider every patient as a potential COVID, patient. So our measure have to be extreme in our cases. So thank you so much for your review. Thank you.
Dr.
So
Akol,
next,
move out from this. You have to stop.
Yes, we're going to move. And next speaker, it's Dr. Jatinder Wahi. Okay, he's the medical director and in the Department of Ophthalmology in Green's Medical Center in India. So thank you very much. You can move on. We're seeing your presentation already.
Dr. Akova, you have to stop sharing the screen.
Yeah, I'm trying to do that. Yeah.
No problem. You have done it. Yeah. So good evening, dear friends. After hearing a wonderful presentation from both Dr. Akova and Dr. Akova, very nicely presented about the finer details and intricacies about this coronavirus disease that is COVID-19. Now, I thought that let me talk something about the medication part, which is now very loudly talked about. There's a big hue and cry all over the world about this molecule Remdesivir, which is being talked about, and President Trump himself called the entire committee into his Oval Room, and there was a big name there, Dr. Anthony Fauci, who is the clinical director of Department of Allergy and Infectious Disease, National University. So he was himself there. And let me start my talk. See, I'm going to give you a very basic concept about what this Remdesivir is, what all things we are hearing about this molecule, how does it exactly stand in present scenario, and how it is going to do good or harm, and what can we really expect about this molecule Remdesivir. Let me start with two very bold statements. One is himself given by Dr. Anthony Fauci. He said that the Remdesivir is a milestone and not a cure. So we have to be very particularly and explicit about this statement. And there is another statement given by Dr. Stephen Parodi, he's the chief lead of control of infectious diseases USA. He said that Remdesivir is speeding up recovery in COVID-19 patients, but it is not a cure. So both these bold statements are ending with the word not a cure. So taking this small words into our mind and with a basic knowledge, let me just move ahead. So exactly what is Remdesivir? Now, this is a proclaimed antiviral drug, and it is basically a prodrug. Prodrug of what? Prodrug of GS 44152. You need not worry about these numbers. So basically, it is a prodrug. That means that whenever it enters the body, it will get activated. And let me talk about some very basic things we have read in our medical sciences a long way back. Our entire DNA and RNA is being made of very basic elements that are nucleotide bases. And you have heard the name, that is cytosine, guanine, adenine, thymine, uracil, all these bases, which are the building blocks of RNA and DNA. So this drug is a adenosine analog. It is a very similar analog base to a nucleotide adenine. Now, this drug is administered IV. It is no more like, let's move about the confusion. I've heard some people, is it available in pill form like the Tamiflu for the swine flu? No, it's not. It's an IV drug. And it was used, it is not a present drug. It has been made very way long back and was used for treating Marburg and Ebola virus by Dr. Richard Besser. He was a chief lead who was working upon it. But it did not work on that Ebola virus. So this drug was kept inside the storeroom. It was there. And now it has again jumped out of the box. And the only company which has been making this Gilead USA company, that pharmaceutical company is manufacturing this drug. So with this basic knowledge, let's move forward. What is the mechanism of action? So now this drug is a nucleotide analog to inhibit RNA synthesis. What do you mean by this? As I've already told you that there are 4 nucleotide bases in cells: adenine, guanine, cytosine, and uracil in RNA. And in DNA, the uracil is being replaced by thymine. So this is present in DNA, and this nucleotide is present in RNA. So when I just have a face-to-face show of Remdesivir with adenine, so it is very nice to see that this nucleotide base is having the same shape as this part of the molecule of Remdesivir. So this just looks like a photocopy. So if I give a Remdesivir to a certain patient, this part is exactly of same shape and size as of adenine nucleotide base. Having said all these things, let us just see the beautiful picture of this super tiny deadly beast coronavirus. Professor Morat has explained it beautifully with all its S protein spikes jetting out from the lipid envelope with a protein coat, and then inside a single very long single-stranded RNA. This is the basic genomic material into this virus, and this is the main culprit. Now let us see how it goes about. So now, as both the professors have said, the transmission is by the aerosol or by the droplet method or whatsoever. So it gets transmitted from one patient to another by sneezing or coughing. And once this goes inside, as professor has already said, that there are ACE2 receptors in the nasal mucosa, respiratory tract, lower respiratory tract. And the spikes of this virus just go hand in glove position with the ACE2 receptors. So this virus comes there, it gets attached with these ACE2 receptors as if there is a hand in glove position. And that is how this is an entry point that this virus enters the host cell. So now this is the cell membrane. This is the ACE2 receptor. The virus is here. It has got spikes. It comes here, attaches here, and it gets an easy entry as if you go to a movie, show the ticket, and you enter the cell. So now you have entered a cell. This is the whole cell. Now this virus, when it enters, now this is a clever beast. So it wants to multiply itself. And for that reason, he just knocks the door of ribosomes, that is the cellular body inside our cell, which has got an enzyme, RNA-dependent RNA polymerase. So this virus very cleverly takes over these ribosomes, enzymes, and this is his RNA material, single-stranded RNA of the virus. Now it takes help of these enzymes and tells them this is the cell, there's a RNA of ribosomes. Ribosomes have been taken over. Now this virus tells that this is my RNA, So now you do not work for the host cell, rather you produce my duplicate copies and reproduce a new viral RNA as similar to the mother RNA. So this is what it does. So it has taken over the ribosomes, the RNA polymerase. This is the original virus RNA, and it orders the host cell ribosomes to make similar copies here. Now, this is the RNA strand, and RNA polymerase is picking up all the nucleotides from the cell, and they are being attached all over. This is the enzyme, and the long sequence of RNA is being made. So this is the mother RNA of the original virus, and this is the child viral RNA, very similar as it wants. So it has hijacked the cellular mechanism, and now the RNA is being made of its own choice. So no more of a original host cell DNA. But now what happens when we give a remdesivir? See, we have given a remdesivir. It also enters the cell. Now this remdesivir's one part cheats the RNA-dependent RNA polymerase. So this part is being cheated, and the adenine base, which was nucleotide base, which was to be used to manufacture this viral RNA, is being cheated, and instead this remdesivir molecule gets attached as a nucleotide base instead of adenine. So adenine was to be picked up from the cell and was to be used to make a child RNA, but that has been cheated, and now remdesivir gets fitted here. When this happens, this virus is very clever. Despite all these things that we have now got a defective RNA, it has got a beautiful enzyme known as 3,5-exoribonuclease. Now, this is a spell check mechanism which the virus has. It's so intelligent so that he can pick up these defects, and it has a power of autocorrection so that he feels that, no, I am being cheated. This is not my original RNA which I wanted to be made. So it gets the defect repaired. So this is the beauty of this deadly virus. So now this is the RNA polymerase, and the remdesivir molecule has got attached. It has cheated this enzyme, and now no more further nucleotides can come and attach here. And then the chain is, these are all being repelled. So the new virus RNA is being not made. And then when the host cell is full up of these viral RNAs, there is apoptosis. The cell bursts, the viral genomic material spreads out, and it goes to take over to the other cells. Now, this is how the remdesivir works. And what are the side effects? As with all the viral agents, nausea and vomiting is one, but the basic important side effect is transaminitis. That is, that it elevates the liver enzyme. It causes damage to the liver cells, and it causes elevation of liver enzymes. So that is an important marker. And moreover, this drug also causes lowering of blood pressure. This is very, very important. So the BP can really fall down. So it has to be given in proper medication under the great supervision. Now, why the remdesivir went into the trial? This is very important to know. The phase 1 was that when it was given to the real sickest patient to get relief, and it showed shortening of illness course as well as statistically significant decrease in mortality. This was the phase 1. And when the phase 1 was being tried, the results were so good that it was decided that let us stop giving placebo to the other patients. The results are very encouraging, and let us give this drug to the other patients also. And that is how the phase 2 means that every patient of COVID-19, every patient will get this molecule remdesivir. So this is how the phase 2 went on. And for this, we had in vitro data. Just remember, we have a data which is a work done in Petri dish. That is, this molecule shows an activity against COVID virus, MERS-CoV, but this is not being proved in vivo. So the trial was going all across the globe in 68 sites, and they have enrolled more than 1,000 patients all over. And then the FDA has shortlisted the situation, and it has given a EUA permission. Now, what is EUA permission? It is an emergency use authorization. So they have modified the FDA rules, and the entire sequence has been little shortened. And seeing the clinical situation of the global pandemic, emergency use authorization has been allotted to this molecule, remdesivir, but with a clause that it has to be used in seriously ill or sick COVID-19 patients. And then there was a situation that how to define who is very seriously ill. So then, according to AAO guidelines, those patients who were having ARDS, those who are needing ventilatory supports or mechanical supports, and those are of the having another comorbid diseases and were really sick were termed as seriously sick patients. Mind you, my dear friends, it is not a cure, and some of the treated patients with remdesivir will still die. So it is not an absolute cure. What does remdesivir delivers? And it has been categorically said and being phrased out in 3 simple words. It gives 31% quicker recovery of patients. This is a bold statement and a very calculated statement. It gives 31% quicker recovery of the patients. This translates into 4 fewer days in hospital stay. That means on an average, if a seriously ill patient is taking 15 days in a hospital, he will get discharged in 11 days. This is what has been calculated retrospectively. And the drug acts in a way that it stops the virus multiplication. So this is how the remdesivir delivers. And if there are less of the hospital stay days, that means less chances of septic shock, ARDS, nosocomial infections, and less intubation for ventilation. So these were the advantages why this drug has jumped a sequence ahead and is now being tried. Moreover, this drug has just given an important footstep for other people to work upon the various other stages how the virus can be attacked. This is one foothold we have got, and now many people will come on, many drug companies will come on, many researchers will go on, so that we can really see how this other factors can be used to stop this deadly virus. Now, is there a reason that this drug can be given to the patients with the mild symptoms? And the answer is big no. No, this drug can neither be given prophylactically nor be given to the patients with the milder symptoms. It is very, very clear. It is not a pill form and cannot be used as an over-the-counter drug. And we cannot confuse it with the tablets just like we had for Tamiflu. There is a very interesting fact from Spain, and the author is Joseph M. Libre in his article. He has seen that quite unexpectedly, people living with HIV, those who are already being infected with HIV, are not at an increased risk of acquiring COVID-19 disease or progressing to ARDS once they get infected with the coronavirus. That means a person who is already living with HIV has got a less chance to get infected. Moreover, if he gets infected, it has got a less chance to progress to ARDS and other serious complications, God knows why. And there is also an association that the risk may be even lesser with the general population when you compare this HIV-infected patients. So there is something, other things going on and on and on, we don't know. But this is a simple foothold which we have received, and it is just a methodology, but it's not a cure. And there is an important information for Indians that Gilead is getting associated with India with 4 companies. This is Cipla, Hetero Labs, Jubilant Life Sciences, and Mylan for making and delivering Remdesivir. So this is a big news for India. And there is one more company which is allotted to Pakistan. So this is how the story goes. This is all I wanted to say about Remdesivir. Thank you so much for your patient listening, and I'm open to your questions. All over to you. Signing out.
Dr. Wahi, I guess Dr. Maria has some connection problems.
Oh, that's no problem. Dr. Agnieszka is there? Yes.
Yes, here I am. That was very good presentation. Thank you very much for it. But could you specify the indication to use this drug? Because from other hand, I don't know if it will be because the indication are not very clear for me. When would you use such drugs?
Agnieszka, you're absolutely right. Indication is not clear.
It's very unclear because it's like left for the judgment of the doctor that actually deals with the patient.
You are very rightly pointed out, and you are absolutely true. Indication is not even clear to me also, and it is not even clear to patients, to all over the doctors all over the world. See, what the thing is that this molecule is being judged by the committee of the people who are dealing with the very sick patients in USA. So a certain category of doctors have been permitted with the EUA permission, that is emergency utilization authorization, and they will be taking the decisions, and they will be soon giving out the guidelines that what do you exactly mean by very sick patients and to whom it has to be given. So that is not very, very clear. And I really appreciated that you made this point out.
Yes, but there are also analysis showing that that actually frequency of very, very severe complications connected with SARS are the same frequency as, let's say, comparing to the flu.
Yes.
So is it justified, for example, to use a special drugs, very probably very very costly, and to, for example, to shut down the whole economy, whole world actually, when we deal with the disease that actually is a little bit like as frequency goes compared to the flu. That's
Agnieszka, you are very right. Now, this is just like a situation of what we really call it as, and what the people in USA have called it as a last ditch stand. That means those patients who are at the terminal stage and there is nothing in our armamentarium, so we are going to use this drug and see the results. So they have done it on more than 1,000 patients picked all over the globe. It was a double-blind, randomized clinical placebo trial, and out of which they just gathered one simple information that we are not going to cure these patients. So now see, we are giving a drug, but we are not curing the patient. The only thing we have reached till point is,
what I'm trying to emphasize is that we are facing very difficult situation, and we in the clinics we have some decisions to make, and we need to do our best because the situation is moving. Okay, we just need to talk one to another and to try to have the as vast experience as possible. You wanted to ask something?
yeah, no, I just wanted to add that we need more clinical double-blind trials. We need more trials, and then only we can need to a certain specific foothold. Otherwise, it's really big trouble. But this is a simple ray of hope we have received till now.
Dr. Jatinder?
Yeah.
I would like to add that the actual results of the last patients were very promising. So the fact that the initial results of Remdesivir of basic investigation with monkeys were much better, but it's everything because patients were given just in late stage of the disease.
Yes.
So when monkeys were given Remdesivir after inoculation of the virus, it was given for 10 days, but just 12 hours after inoculation. And that made a big difference. So the thing is the results of the actual mortality with Remdesivir, that is 8% compared to 11.6%, and the discharge time of 11 days compared with 15 days, is very promising. And we think that moving into treating earlier patients with, we still have to know the phenotype of the patients or the characteristics of those who are going to go badly. But treating early with Remdesivir may be the key. So it is a very, very promising treatment, but we still have to know a lot of things.
2 papers are coming where they have started giving for little earlier phase also, but still
Exactly.
So it is still in dark,
Exactly.
All over to Maria.
Yes, I don't know if someone wants to point something else. Well, if the next speaker is Dr. Rajesh, it's a medical consultant in a cornea and refractive service in Aravind Eye Hospital in India, and he's going to talk about innovations for COVID management in an ophthalmic practice.
Thank you, Marian. Due to COVID pandemic, the whole world around us has changed. Let us see some of the changes which we have done to make the ophthalmic practice into a more safer one, both for ourselves and our patients. I am Dr. Rajesh from Aravind Eye Care System. Aravind Eye Care System consists of 7 tertiary care centers catering to 1,000 to 2,000 patients, 7 secondary care centers catering 150 to 400 patients, 6 outpatient centers, 85 primary care centers. In a single day at Aravind, we see around 13,000 patients, 1,500 surgeries are examined, 5 to 6 outpatients camps are conducted, in which 1,500 patients are examined. 300 patients are transported to base hospital for surgery, and classes are taken for 100 residents and 300 technicians and administrators. This is before the COVID pandemic. And after the COVID pandemic, only the tertiary centers are functioning, and 1,500 patients are examined each day. We do 75 surgeries a day. Outreach camps are suspended, and online classes are taken both for the residents and MLOPs. Suddenly, the nation announced a total lockdown. We had restricted transport, break in supply chain, so we are not able to procure any goods. And the safety is of utmost priority. When we are examining any emergency patients, we should make sure that we give PPE for all our staffs, and even the procurement of which is very difficult. Coming to the COVID transmission, it occurs via two methods. One is touch. When I say touch, it not only means direct touch, but also by touching the handrails, doors, switches, and also while handling money or credit cards. The other method is aerosol and microdroplet. So it doesn't only spread by coughing or sneezing, but it even by talking. So this video shows small microdroplets which are generated while talking. And these microdroplets stays in the air nearly for 2 to 3 hours in a closed environment. So to prevent the COVID transmission, we should protect ourselves from both these things. We should protect the doctors from the patient. We should also protect the patients from the doctor, because any one of us can be a asymptomatic carrier. And also we should make sure that these COVID doesn't spread among the patients who are coming to the hospital. This has to be done in a dignified manner, so the patients are always feeling respected. As Winston Churchill rightly said, never let a good crisis go to waste. We must look at crisis as an opportunity to better our ophthalmic practice. So we used what limited supply we have and came up with novel solutions to a lot of these problems. Coming to system innovations. Layout modification. First, we made sure that all the entrance to the hospital is closed except for one, and all the patients and staff enter or exit the hospital via this entrance. No air conditioning system is used. Windows are kept open for ventilation, or window curtains were removed. All the doors are kept open to reduce patient and staff touch. All unassigned areas are completely shut from patients and staff access. Triage. We made sure that we measured temperature for all the patients coming to our hospital. And even while measuring the temperature, social distancing is maintained. We also made sure that we get a COVID consent form from each of the patient, asking them relevant questions. All the patients coming to our hospital were screened for conjunctivitis, because conjunctivitis is one of the symptoms of COVID-19. And to examine these patients, we designed a conjunctivitis clinic. This video shows how it is done. We just remove the chin rest from the slit lamp, and in the plastic sheet, we made a small slit-like opening, and the chin rest is fixed to the slit lamp while sliding in this opening, thus achieving an airtight chamber. The plastic acts as a barrier between the doctors and the patient. This video shows how patients with conjunctivitis are examined. This gives us total protection. Coming to modified examination method, we made sure that all the examination are done in a well-ventilated area. We have changed the examination of vision and refraction from a closed cubicle into an open area which is well ventilated. We have also put a scribe sister both for the vision sister and also the refraction area. So the person examining the patient doesn't contaminate the keyboard or the desk. So the area where the doctor examines the patient is also very well ventilated. They examine with a face shield, respirator, slit lamp shield, and we keep a fan behind. So once the fan is switched on, it blows away all the aerosol away from the doctor. We also keep a scribe to enter all the findings. So the examining doctor doesn't contaminate the table or the keyboard. We also made sure that when you talk to the patient are being seated a little bit away and not in front of the slit lamp. We found that while examining the patient with a 90 diopter lens, we invariably touch the mask of the patient. To avoid this, we have done a fundus photographic cubicle. Here you can see the plastic sheet is cut and the foot cover is separated into two parts. The one end of which is stuck onto the plastic cover and the other end to the nozzle of the fundus camera, thereby achieving an airtight chamber, while we are able to move from the left eye to the right eye while capturing the image. The patient comes and after being seated, he shows the MR number in the MR card. The fundus photography of the patient is taken by a non-mydriatic camera. After capturing this image, this is transferred to the electronic medical record via ERCOD. And the doctor opens the electronic medical record and directly sees these images. They don't have to image the patient with a 90 diopter lens. Coming to the behavioral innovations. So we made sure that we put a wash area in front of the hospital. So all the patients and staff who enters a hospital washes their hand with a soap dispenser and tap. We have made it a foot operated soap dispenser. So they don't contaminate the nozzle or the tap with their hands. And we incidentally also found that this saves a lot of water. And we feel that in the future, all of the taps all over the world will be switched into a foot operated one. We always insist on the use of hand dispenser. But while touching the nozzle of the dispenser, we always touch the nozzle and contaminate it. And this may itself be a source of transmission of the COVID virus. And it stays in the plastic for 3 days. So we have designed a foot operated dispenser. All we have to do is just press with the foot pedal of the dispenser while keeping the hand below the nozzle, achieving a hand-free sanitization. This was done using a make-it-yourself reverse engineer method. This can be easily done by anyone, and the instruction of it is being uploaded in the Oro tube. This doesn't have any springs or electronic into it. So just pressing the foot pedal will make sure that you achieve a good sanitization of our hands and also avoid contamination of the nozzle. Though sensory-based sanitizers are available, those are very costly and difficult to procure at this time. And also they malfunction a lot of time. Registration cubicle. Initially, our registration cubicle was open and the patient used to touch the front desk, which may itself be contaminated and be a source of infection. So we have put a proper registration cubicle here with a small slit-like opening for giving the cash and getting the MR card. We also put a speaker system for seamless communication between the patient and the sisters. Waiting chairs. We made sure that we achieve social distancing by tying a band around the central chair. But the patient used to keep their bags or bottles over this chair. As behavior of the patients can't be changed, we have removed these central chairs, thereby achieving good social distancing. Coming to technological innovations. In the initial period when the lockdown was announced, all of us wanted to use a face shield, but it was very difficult to procure. So we did a make it yourself face shield, where the OHP sheets and a sponge is used, to which a double-sided cellophane tape is applied. An elastic band is cut and the foam is stuck onto the OHP sheet. The elastic band is fixed via stapler pins. This is very easy to make. Now, at present days, you can get a lot of shields, but 6 weeks back, it was very difficult to get them. We have also made a stand for this face shield. Most of the time, we keep the face shields down and it gets scratched, decreasing the vision. So we made sure that we always store it here. And you can see clearly that all the doctors can name their own face shields. At the end of the day, this is taken, sterilized using a UV chamber, and then given to the doctors the next day for use. Slit lamp shield. We have also put a slit lamp shield to protect our doctors from the patients. We have also labeled with a blue and yellow sticker for fixation of the patient, which makes our examination very easy. 3D printed masks. When the N95 mask was very difficult to procure, we tried to print a 3D printed mask. So we got the design from the open source called Thingiverse. We 3D printed it, and we were able to fix a normal filter or a N95 filter to this 3D printed mask. And at the end of the day, after use, we can take away the filter and sterilize it using a normal solution. Mm-hmm. We have also tried 3D printing a shield, but as the filament was very less, we were not able to 3D print the shield for everyone in our hospital. History over phone. Once the patient registers their card and comes and sits in the waiting area, the doctor calls the patient over phone and gets their complaint and history and write them over the EMR. This drastically reduces the amount of time the patient needs to spend in front of your slit lamp, and the talking time is also greatly reduced. So we can directly start examining the patients. This further reduces the risk of transmission of COVID while examining the patient. UV freezer box. We converted the normal freezer box into a UV light fitted freezer box for sterilization of the respirator. At the end of the day, the doctor names their mask and put them in a paper cover. This is collected from each doctor and these are placed in the freezer box. So here you can see a sister collecting all these masks, and this sister used to be in full PPE. She removes the mask from the paper cover and hangs the respirator in the UV sterilization box. We have also placed a glass behind the box to reflect the UV light for proper transmission. We also spray hydrogen peroxide 3% and we give a 20-minute cycle of sterilization. After sterilization, these are put in the named pouches. Here you can see the doctor's name, the date when the respirator was issued, and the number of times it is sterilized. And these are kept in separate boxes. Each day, the doctor or the paramedics comes and take these masks, and they use their own mask, which is sterilized using UV light. We have also sharing our ideas. We have kept a lockdown Jugaad series where the doctors all over India are sharing their innovative ideas to combat COVID. All of this are recorded and put in OroTube. And all the innovations which I have also mentioned are put in the OroTube. This Saturday is the finals of all these innovations. You may join them. I would like to end my presentation by giving a quote of Dr. Govindamba Venkatasamy, who is our founder. Intelligence and capability are not enough. There must be the joy of doing something beautiful. Thank you. I thank Biotech and Dr. Venkatesh for giving me this opportunity.
Thank you very much, Dr. Rajesh. That was actually pretty impressive. You guys have really smart people in India. I think we should all learn from you. I mean, I've been really impressed. Seriously.
Initially, we were facing a lot of difficulty. We are not even able to acquire these pipes for doing this dispenser because all of it was locked down. So we are not able to get anything. So we thought and we came up with innovative ideas to combat everything.
Yeah, but you guys are smart.
Can I ask
We have a separate task force. So whatever ideas we have, we used to share with them. And all these task force work as a team to get it done within a day. And this dispenser idea was done within 2 hours. So we have a very good team. It is all a team effort.
I see. So now
I think we want to talk.
Yes.
Yeah.
Well, now we have the questions if you want, or someone wants to point something. Well, I can point something about. Well, maybe we didn't say it, but when you consider and when you see, and I can ask this to even Dr. Yonka or Dr. Rajesh. When you see a patient with an acute conjunctivitis, what do you make? You make that person wait in a different waiting room, or they wait in the same area? What is your protocol?
We screen the patient even before the registration. And once the patient is found to have conjunctivitis, we take them for a separate conjunctivitis clinic. We have a separate clinic which is different from the normal flow of the patient. So we don't expose because these patients can be COVID positive patients. So we don't want to expose these patients to other patients. And we design this conjunctivitis clinic specially for them. So we take them separately to the clinic, we fast track them, we examine them, and we make sure that we send the patient off early. So we don't make the patient wait.
Okay.
So
What kind of surgery?
I have got one question for Dr. Murat. Can I ask?
Can I ask the question, Dr. Rajesh, please?
Yeah.
Because I was wondering what kind of surgeries do you perform at the moment? Do you do only emergency surgeries, or you kind of tend to go back to the normal?
Still now we are doing only emergency surgeries,
Okay.
but we are planning to start the normal surgeries in a few more weeks' time.
Okay, but if
till then only the emergency surgeries like open globe injuries or phacolytic glaucoma.
Yeah, I understand. I'm in the retina field, so pretty much most of my surgeries are emergency, so I do not have such a problem. But I was wondering, do you think of any procedure that you could employ? How would you assess the patient? Would you like to screen everybody for COVID and then allow to, for example, cataract surgery? How do you plan to do it?
So,
or you just treat everybody as possible COVID patient? Yeah,
that is the right decision to make, because we can't rely on the test, and getting the test itself is very difficult. So, yeah, and it gives a lot of false negative also. So treat every patient as a COVID positive patient. So we should take all the precautions, and after we should also be in a full personal protective equipments. So as we do as COVID positive patients, and at the end of the surgery, you make sure that we sterilize the whole OT before starting the next case, and also we give a time interval of 20 minutes.
Okay. Okay, thank you.
Maria, I have got two questions, one for Dr. Murat and one for Rajesh. Can I ask them?
Yes, sure.
Dr. Murat, Professor Murat, Is there any role of these ELISA tests, IgG test or IgM test? Do they hold anything in present scenario?
Well, actually, it's a part of the diagnostic armamentarium, actually. But the problem is, when you're doing the IgM and IgG tests, it's a sort of late coming tests, because, I mean, if you look at the IgM usually turns to be positive after the first week, and IgG is a little bit later. And IgM actually wanes and disappears within 4 or 5 weeks, and then IgG continues. But another problem is that not all of the patients will develop these IgG and IgM bodies. So one can expose to the virus, get it, and most of the time these people are asymptomatically developing disease type of patients, and they will never develop antibodies. So these tests, I think, can be used for screening purposes, but not for the diagnosis. So in your practice, for example, if you'd like to operate on a patient, you cannot rely on the IgG and IgM. I mean, well, if the patient is IgG positive, for example, that's a sort of safe type of situation, because that means the patient has been exposed and developed antibodies. But still, we don't know exactly what are the sensitivities and specificities of these tests. So this is still in the stage of development. So at the moment, I would prefer to do the PCR. And now there are some antigen tests emerging as well. But then, as already mentioned, these tests are not highly sensitive. So you cannot rely on a test which becomes negative, and then you can operate these patients freely. I think you should take every measure, even though the patient test is negative.
Okay.
There's one more question to Dr. Rajesh.
Yeah, please.
We are, as per our discussion in this panel, we are just talking about keeping the rooms open, staying apart, social distancing, and all those things. But when we operate, we do operate in a closed modular operation theaters with a running AHU system where the closed air is being circulated all the time. So what is the take on that? Should we just open the doors of the OT and let the AC run, or we should not use the air conditioning, central air conditioning at all while we are operating?
Our OT is designed in such a manner that it is closed to avoid the infection from outside also. So we can't take chances there to keep it open. Better to have a personal protective equipment while you operate it. At the end of the surgery, you can open it and keep the AC running for at least 20 minutes. So the whole cycle, make sure it washes the virus away, virus load away. And a laminar flow is also needed for washing it away.
Okay.
But still we should come to a consensus over it.
Okay, I would like to put a question to make a question to Dr. Amsalem. Well, we know that chloroquine and hydroxychloroquine have been used widely. Do you test these patients for retina toxicity? Do you do any kind of visual field or OCT? Do you test? I mean, do you control these patients?
Well, the daily dose given to these patients is pretty high. 3 times, 4 times the usual dose. However, it is only for 10 days. So the cumulative dose is pretty low, and we believe we don't have to do the follow-up of those patients. What we are really concerned about this whole thing, and let me ask questions, is that we are having a lot of visual casualties because of the COVID spread. Even maybe ophthalmology is the specialty that has been the most affected by lowering his activity, and we have to take it back. I think we have to consider every patient as a possible transmitter and do all the precautions, but start giving our practice, as Dr. Rajesh, with the maximal measures. I would like to point something about ozone. Ozone delivery machines are pretty affordable. And if you put a concentration of ozone at 1 ppm for a few minutes, you may be able to sterilize all the whole place. So it is very interesting for doing it at the middle of the day or for the night, and you can even program them. They are very affordable. We are talking about ozone delivery systems of 30 euros or 100 euros, and you can get 1 ppm, even 2 ppm if you want. And actually, the authorities in Spain are using it to for the medical transport sterilization, for the public transport of the buses and taxis, and also for military transport. So ozone producers may be interested in lowering the virus charge. Thank you.
Interesting. I don't know if well, Rajesh was talking about all these innovations. Have you tried ozone or? I don't know if you can hear me.
We at the moment in Poland, we have some companies that actually are selling some sort of sterilization using the ozone. That's something that was not present before. That's what they do. That's new business, for example.
Yeah,
I guess.
But it's a very
okay. I don't know if it's...
Excuse me.
Yeah.
Excuse me.
Yeah.
Yes, I'm very sorry because I was in the OR and I couldn't be able to be sooner with you. But I was worried about maybe the ocular presentations of the coronavirus, of course, the conjunctivitis, but as the many autopsies performed in a bigger number now in patients with advanced coronavirus that had died, are showing that disseminated intravascular coagulation is the main problem causing to death. And we know that deep disseminated intravascular coagulation in ophthalmology gives bilateral serous retinal detachments, choroidal hemorrhages, and other manifestations, also optic disc swelling or CME. So we may see in severe patients recovering from this disease, those discharged from the intensive care units, some of these findings or the relapsing findings of those damages in the choroidal vasculature and in the eye vasculature, as it is happening in the lungs or the brain or the heart.
Well, I think Dr. Yonka showed an article showing that they have already seen some injuries in the retina, right? And this Brazilian.
Yes, there's a very recent publication from Brazil, São Paulo actually, in Lancet, showing retinal hemorrhages, cotton wool spots, and some deposition on the OCT on the retina. But it's only 12 patients. But it's, I think, as long in the coming days, we are going to have more experience on that. But there's nothing on serous detachments, but it could happen in severe cases in intubated patients. For conjunctivitis, it's in big, I'm very much impressed with Aravind Hospital setting, but in practicality in our life is we have a separate room for conjunctivitis, and we see the patient and we question now for COVID systemic findings. Although even if there isn't anything, we see the patient. If we suspect COVID, we do the PCR. Otherwise, we want to send the patient right away as soon as we solve the patient's problem.
Okay, perfect. I don't know if someone else would like to ask something, or we can go with the questions.
Maria, I would just like to address Professor Murat once again, just for a moment.
Sure.
Professor Murat, seeing the precedents of the past time, I was not born that time when the Spanish flu was there. And it has been widely talked about that there was a second wave, which gives really chills in my bone and spine. So is it a possibility that we might have a second wave or a second spike of coronavirus in this present scenario, sir?
Well, one of my last slides actually was related with this question, and it is expected, of course, but there are different types of scenarios. So, and it depends on the type of immunity the patient is going to develop, and the frequency of the herd immunity is going to develop in different populations. So in the best case scenario, up to 60 or 70% of the whole population develops immunity due to the disease or the vaccine, an effective vaccine. Then you can expect an effective herd immunity develops, and there's not going to be any other wave. So you eradicate basically the virus. But we can foresee that this is not a near future possibility. So we cannot expect that 60% of the population suddenly develops immunity to this disease. It will take time. And there is no vaccine in the horizon for very near future. So it depends on the prevention on the public level, that you keep the distance, these safe distance, social distance, and also the effective measures that you can identify the effective, the infected patients and the case contacts, and you isolate them. So it depends in a given population how effective these measures are. You can prevent a second peak, but otherwise, like what's happening in Turkey now and in United States now, the public has started to go out and the lockdowns has been removed. So I think a second wave will be unavoidable. One point is that because we are going to, in the Northern Hemisphere, we are going to be exposed more sun, which is UV light at the moment, and the virus is highly sensitive to UV light. So there have been some studies, and there is a one in the Med Archive at the moment from Japan, and it says that the virus is highly sensitive to UV and also high pressure and the heat, the increased temperatures that might prevent the spread of the virus during summer months. But once the climate changes, I think the virus might come back if we don't take the measures properly.
Okay. Thank you.
Okay.
Thank you.
So would you like to make any other questions? Do you want me to, I can go then to the questions that have been posted. Well, actually, yesterday, Sidat Hasnani, sorry for the pronunciation if it's not right, posted a questions to Dr. Murat Akova, and was kind of to if it's really justify all the lockdown. I don't know if that information is right, but he thinks that should it be that justified because the mortality rate is close to the flu. I don't think if that's right. I guess I think the mortality rate is higher than flu. So is it really it is really justified? So
well, obviously the mortality rate is higher than flu. I mean, the flu mortality is less than 1%, and this disease, although the mortality varies from one population to the other, one community from the other, now we can say approximately between 2.5 to 4.5% of mortality, which is pretty high, and this is definitely higher than flu. And then, of course, this disease is incomparable with flu, because for flu we have an effective vaccine, although no, it's not 100%, varying between 40% to 70% effective vaccine, and also we have effective measures to treat flu. We have an antiviral or two antiviral diseases which are highly effective against flu, which can be used for treating the patients. And for this disease, we don't have an effective treatment, given the fact that the Remdesivir can help, but we still don't know about that. So I think the lockdown is definitely justified at the moment.
Justified. Okay. Okay. So the next question, it could be, it doesn't say for who is, but I guess Dr. Yoka ID can maybe answer to this. And Dr. Ayushi Shina is asking, why is the protocol being followed for preoperative cases? Mandatory checks or tests before taking the patient to OR, PCR antibody testings are not possible in many countries. So what do you do in those cases if you cannot do a PCR or an antibody test?
As mentioned before, 80% of the COVID patients can be asymptomatic, no signs. That's why we have to accept every patient possible COVID patients. That's why we have to take every measure to protect our personnel and everybody. That's why we have to use every protective measure, including masks, if we are going to use general anesthesia and 95 masks, and also clean areas. We have to take patients in and out quicker. We have to clean the area, and we have to put time points more than usual. We are not going to operate many patients as we did before. We have to make our schedules more seldom, I would say. I mean, I don't know how the other surgeons, especially you said you operate all the time during COVID emergency cases.
Okay.
I do. I actually, I own my private practice, and my specialty is retina. And from the beginning of the COVID pandemic, we kind of closed for a day or two, maybe, because all the time people were coming. We have 60, I think 70% of my patients are one-eye patients, and they are on anti-VEGF treatment. We had cases with retina detachments or injuries and I observed that the frequency of our injuries went high during the lockdown because people were bored and they were doing stupid things at home. So they put strange objects into the eye. So, but for example, as far as an anti-VEGF is concerned, we were trying to shorten the time of the visit. I tend not to do, let's say, observation. I call patients to the clinic when the treatment is necessary. And for example, sometimes I kind of do treat and extend, or I tend to go more to the fixed mode of the injections. I had also a few patients when I decided to go to make injections at home, which is not basically what I do on everyday basis. But for example, one patient broke his leg, and he was one-eyed patient, and he was not admitted to the hospital due to the COVID. So he was lying at home, and because he was one-eyed, he could not go for the visit. He could not come to the clinic for the injection. So the situation was quite scary for him, because despite a broken leg, he would end up as a blind person due to the epidemic. I can say from my point of view that despite a very difficult medical situation, we also experience lots of trouble with logistic and just everyday life, which add to the very difficult situation. So, but it's getting better, and I think that we kind of used to live with this virus. And of course, I think there will be some sort of new approach to the patients and to dealing with the patients. We needed some time to adjust to the new situation.
Yes, of course, we need to find a new kind of normality.
Yeah,
it's going to take some time.
New normal, I think. That's what we are going to have in the end of this pandemic.
Okay, so there's another question Sorry, my pronunciation, it's a really long name. It's Chinchal Bahwati. Is asking, it doesn't say the question I guess it's for Dr. Murad. Sorry, that was not the question. Sorry, it was from Carlos Chua. I guess it's for Dr. Murad. And it's asking, since HIV patients are taking antiviral medications already, could this be a reason why they react less, or have you heard about this? I mean, HIV patients are already taking their medications. Are they reacting less to the COVID?
Well, I'm not aware about this this type of epidemiological data, but obviously there is one drug which has been used for treating COVID-19 patients. It's ritonavir-lopinavir combination. Although it has not been proved to be an effective drug in many parts of the world, including my country as well, this drug has been used for treating patients, particularly given to those who have been admitted to the ICU. There have been some reports, actually, it might be effective, but there are reports on contrary this. So theoretically, that might be the situation. So let's think of a patient with HIV who has been on ritonavir and lopinavir combination, and this drug can have some preventive activity against COVID-19. This is a hot issue, again, which has been debated, but still there isn't any solid data. The same applies to hydroxychloroquine. As you may know, in several countries now, hydroxychloroquine have been tested for medical personnel as a prophylactic measure in the healthcare setting, but no data have been released yet. So I think this is the same situation for the HIV cases who have been on ritonavir, lopinavir as well. I haven't seen any data yet.
Okay, so next question, thank you. This one, yes, this is from Ching Chok. Well, I might answer this question, and it's asking why Schultz tonometer can be used. Actually. Actually, what has been said about tonometry is what I already said. The air tonometry, it's not advisable because of this pressure from air that can spread all these aerosols. And it has been advised not to use the Perkins tonometer as well, because we have to get really close to the patient. As far for the other tonometry, as far as we can clean all the surface correctly, for example, with alcohol 70 degrees, they can still be used, because we still have, we need to measure our patient's pressure sometimes. I mean, but as far as I know, what has been said is that it's not advisable to use the air tonometry and the Perkins. Mm-hmm. I think there's another question related with pressure. I think with that answer, both of them have been answered. And there's another question from Mutuku Maran to Dr. Rajesh. If you wanted to perform cataract surgery, what is the protocol we need to follow in the OR? I think this has been answered already, maybe. Okay. Yet Santiago Gatmaita wants to ask to any of the speakers, in the emergency cases like sight-threatening disease, where either antibody testing is not available or will delay treatment, if we need to wait for results, is it safe to just assume that it's COVID positive case and just take necessary precautions? I think that's have been answered too. And there are a couple of more questions. Dr. Ayushi Shina. Thank you, Dr. Noblieska. I'm saying it right?
Yeah,
I'm sorry. Spaniard has huge limitations with this kind of last names. I'm sorry. I am a retina surgeon too. Have been working exactly like you have been, taking all possible precautions and with prayers too, Thanks a lot, panelists. Oh, this is a great thing. The panelists and speakers, great webinar. Stay safe, everyone. And the last one is from Bharat Raju. It says, if patient have corona, is it affect the vision? I mean, if they see less, well, it depends on. We're saying that it's not really frequent to have an infection. If they have it, it's usually conjunctivitis. So conjunctivitis by itself sometimes can affect the cornea and cause.
I think that we still have we will have a delay for learning all the manifestations of the coronavirus in the eyes and the complications of the possible treatments for corona for the eye. However, the drama actually is the delay in treating our current patients. Cataract surgery has dropped to 99.7%. That may not be a big problem, but there are cataracts that must be operated. However, glaucoma surgeries have dropped in some studies of 80%. And in retina cases, we know in the clinics, you may have noticed as well, even if I also have been operating a lot of retina detachment these days and injecting a lot of eyes. Patients were afraid, and a lot of patients with intravitreal treatments didn't come to their appointments, or even their family didn't bring them. So we will have to face very big difficulties now, because we have to treat those delayed patients and not to delay, the treatment. So the effort will be big, and I think we have to treat every patient as a possible COVID patient and treat all the eyes that we can treat about those diseases. However, for many consultations, we have to encourage the telemedicine and to discourage some visits, of course.
Yeah, that's right. That's people still is afraid to come to visit, even if they cannot see. I mean, and that's a big problem, because we're going to face patients that should have been treated weeks ago. So, well, let's see how it goes, and hopefully, I don't know if we will have some more peaks, or, well, we will see. We're facing, as I said, a new normality, but we're going to face problems too with the patients that were not treated, right?
Yeah.
Well, there are no more questions. Actually, I don't know if any of you want to point something else, to make any comment, or we can conclude the webinar.
We should meet in some time and try to exchange our experience and to share what our ideas are, because what I notice is that my approach to the situation changes as the epidemic goes. Because usually at the very beginning I was kind of frightened because nobody knows what to accept, and now we are more relaxed and we know how to deal with the situation. So probably it will change again in a month or two.
Actually, every day.
Yeah, I didn't want to say that, but yes, it is true, every day. Yeah, that is true.
Okay.
Entering into a mental equilibrium, the scenario is changing, our mental mindset is also changing. So we are entering into a mental equilibrium, and I hope so, in the name of God, everything will be fine in the coming days.
Okay.
Hope so.
All the best for you. Yeah.
Okay, so thank you very much. Thank you, Biotech. So hopefully we prayed this to finish someday. So thank you, everyone.
Thank you very much. It's a pleasure. It was a pleasure to meet you and to be with you today. Thank you.
Thank you.
Thank you.